Thanks, Bea. Thanks to all the previous great speakers. Sorry to disappoint you guys. I have no incriminating pictures of any of you here. So today I'm going to be talking about two studies which focus on integrating complex and personalized data to study brain development. So the first study will look at integrating imaging and behavioral data, and the second study will follow up on Nico and Damien's talks on personalized networks. All of this data comes from a study conducted in Philadelphia, the Philadelphia Neurodevelopmental Cohort, which was led by Raquel Gurr in our group. It's funded by NIMH and includes over 1,600 kids who are ages 8 to 22 who are all scanned on the same scanner without any hardware upgrades over a 2-year period. This first study I'm going to be talking about was recently published and is led by a truly remarkable MD-PhD student in the lab, now Dr. Cedric Shia. And it focuses on integrating high-dimensional behavioral and imaging data. And this study responds to one of the fundamental problems in psychiatry. I'm a clinical psychiatrist, and when you come see me in clinic, I will be very nice, but I will usually order no diagnostic tests that will direct management. And this is because we currently have no biomarkers which assort with clinically meaningful outcomes or treatment response. And this is thought now to be likely because the DSM does not carve nature at the joints. So the goal of this study is to try to understand the association between clinical symptoms and functional network data in a completely data-driven fashion. This study also responds to how most psychiatric neuroscience is conducted. We get a lot of imaging data, we get a lot of clinical data. However, if you look at the papers we publish, usually we take just a slice. We get a lot of clinical data, but we just look at the depression measure. We get a lot of imaging data, but maybe we just conduct a seed analysis. So the goal that Cedric pursued here was to try to integrate high-dimensional imaging data and high-dimensional clinical data in a data-driven fashion. And to do this, he used a recent update on an old statistical technique. The old technique is canonical correlation analysis, and the update is sparse canonical correlation analysis. It's a regularized version that was developed by Daniela Witten at the University of Washington. And what it allows you to do is it allows you to look for linear combinations of functional connectivity data that predict linear combinations of clinical data. And what comes out of sparse CCA is something not unlike what comes out of a PCA. It's like a scree plot, except on the y-axis you can see here is the covariance explained. And just like in a scree plot of a PCA, you can look for the elbow, and we can look for the components that explain the most covariance. Here there are 7, which are marked on the inset there. On those 7 components that come out, we conduct permutation testing, correct for multiple comparisons with the FDR, and there's 4 components that survive this correction. I'm going to call them mood, psychosis, fear, and externalizing behavior. And the next couple of slides, I'm going to unpack that and tell you why I'm calling that. So this first component, which we call mood, what you can do is you can plot the brain connectivity score on the x-axis versus the clinical dimension score on the y-axis. Again, this is the linear combination of brain features compared to the linear combination of clinical features that are found in a completely data-driven fashion. And you can see there's a tight relationship between them, R values of around 0.7. However, I want to caution you not to interpret these R values as like a standard parametric effect size here, because machine learning techniques are really good at finding signal, and if you look in the inset, the null distribution isn't centered around 0, it's centered around 0.55. However, these are still highly significant, way outside the null. And what you can do is then you can look at what's the highest weighted clinical feature is among those linear combinations of clinical features. And that'll give you a little bit of intuition into what's going on with these components we found. So the first one, the highest weighted feature, which we're calling mood, the highest weighted feature in the mood dimension is feeling sad. All right, that's fairly interpretable. The second dimension, the highest weighted feature is auditory hallucinations. The third dimension, the highest weighted feature is fear of traveling. And the fourth dimension is trouble following instructions. So, however, I've just told you what the highest weighted feature is. We can go beyond that, and we can conduct bootstrap resampling analysis to see what features are significantly weighted in each of these dimensions. And the way Cedric's displayed the data here is on the outer ring, we have the DSM in the colors, in the solid colors with the, can I do anything here? There we go. In the solid colors with the individual clinical items. And then in each of the inner rings, we have our data-driven dimensions. So this first ring labeled A is the mood dimension. And the second ring labeled B is the psychosis dimension. And what you can see in the mood dimension, if you go down here, you get the highest weighting again is feeling sad, but we also get significant weightings in other things in the depression section of the clinical interview, like crying, anhedonia. Also, we have significant weightings outside the depression section, like from the OCD, but it's thoughts of harming yourself. In the second dimension, we have the highest weighted feature again is auditory hallucinations, and mainly significant weights within the psychosis section of the interview, but we also have significant weights in the mania section. So what you see when you look across these dimensions is these completely data-driven components that are pulled out of the data align with our clinical experience, but also bleed across the categorical diagnoses that are codified in the DSM. You can do the same sort of analyses for the functional connectivity data. There's a lot of awesome stuff in this figure, but unfortunately I don't have the time to get into it. But one thing I want to emphasize is that each dimension has its own specific pattern of functional connectivity, but there's also commonalities that go across dimensions. In particular, one commonality that's present across all dimensions is a loss of modular segregation between the default mode and the task-positive executive system. And why this matters is because we know this is one of the most fundamental and reproducible effects that we see in brain development. It was actually initially shown by that guy, Damien Fair, back in 2007 in PNAS. We replicated it in the PNC using functional data, and Graham Baum, an excellent PhD candidate who's about to defend, and I think sitting right over there, recently showed it in that the same thing happens using structural networks defined by diffusion imaging. So what we see is that normally in the adolescent brain, what happens is that networks become more segregated, but a common feature across all these data-driven dimensions is this pattern of modular segregation is lost. Now, one thing that I should have emphasized at the beginning is that anytime we see something, I'll believe it a lot more if you show it to me twice. So all the data I just showed you was from a discovery sample of 600 some people. We left out a replication sample of 336 people that we didn't touch. And then Cedric went back and applied the same model without any parameter tuning, and most of the results replicated, not all of them. In fact, the psychosis dimension did not, in fact, replicate. It's a smaller sample size, it can happen, but the other dimensions did, suggesting that these are reproducible data-driven dimensions linking functional disconnectivity and clinical symptoms. All right, the next section, in the remaining time I have, is unpublished work from a fantastic postdoc in the lab, Zaishu Kui, who's here at the conference and will be presenting data. And it focuses on some of the work that Nico and Damien were presenting earlier and applies it to development using data from the PNC, looking at personalized functional networks. So this project responds to the limitation that they really clearly laid out, which is that last project I showed you, Cedric's project, all the other studies we've done in our lab use a group atlas. However, there's been a rapid accumulation of very convincing high-profile papers across multiple independent groups and data sets showing that functional topography is quite variable in humans. And what I mean when I say functional topography, I mean the spatial layout of functional networks on the anatomic cortex. Now, normally, when we process functional imaging data, we take the functional images, we co-register them to the structural image, and then we use the fine-grained structural information to warp them to a template. This process, though, assumes that lining up the structural images will line up the functional networks. And this data shows that that's just not true. So there's a lot of very convincing data from Nico, Damien, other groups, Janine Beisterdorf, who's here, Sheng Liu, Thomas Yeo, who have shown that there's marked functional variability there's marked inter-individual variability in how these functional networks are laid out in humans. Furthermore, this variability itself is heterogeneous across the cortex and is maximal in association cortex, in particular, the frontoparietal control network, which is responsible for executive function. However, despite this data from adults, it really hasn't been shown, to our knowledge, how this functional topography evolves in development and associates with executive function, which we might think is important because the same systems that are responsible for executive function are those that are most variable. So to look at this, we use data from the PNC, and we use 27 minutes of data here. And this is important because, as Nico and Damien were saying, data quality and quantity is very important for this. Here, to get to 27 minutes, because we only had a short resting state run, we used not only the resting state scan, but also task-regressed task fMRI data. work from multiple different groups, so shows that's a reasonable approach and allowed us to get more data quantity here. And to define individual networks, we used a non-negative matrix factorization-based technique developed by our excellent collaborators at Penn, Hongming Lee and Yong Fan. And what's nice about this non-negative matrix factorization-based technique is it allows you to specify any arbitrary number of networks. Here we chose 17 to allow comparability to other techniques. And it gives you discretized networks, but also at each of these networks, it provides a continuous loading of each network at each vertex. And so this technique starts by giving you a group atlas. Then that group atlas is iterated and adapted to each person's functional data. And we started by simply looking at individual subjects' data. And what came through is that though there's clear correspondence, which is enforced through the use of that group atlas that is then iterated to each individual, there's a lot of differences in both when you look at the discretized networks and the fine-grained patterns of the loadings. In our observation, this was highest in frontoparietal cortex, which you would expect from the adult data. We can quantify that by looking at variability and parcellation across the whole groups. And you can see that indeed, just like in adults, when you look at kids ages 8 to 22, there's the highest variability in association cortex. And if you summarize it by the functional network, you can see that this variability is highest in frontoparietal cortex and the ventral attention network. So as a next step, we wanted to see, well, we know these networks are variable. How do they evolve in development, and how do they associate with executive function? And to look at this, we started by defining a really simple summary measure, which we call the total network representation. It's created by simply summing the loadings of each network in template space. And it's important that we do it in template space because this prevents aliasing in any surface area effects from known anatomic changes that occur in development. And what we did is we simply looked at how the total network representation of each of our network changes with age and then associates with executive function while controlling for age. And somewhat to our surprise, the total network representation, how much of these networks were present on the cortex, didn't change with age, but was associated with executive function. Additionally, it was pretty specific in terms of the associations with executive function. It wasn't any network. It was just the frontoparietal networks and one of the ventral attention network. So as a next step, we went beyond that extremely simple representation and looked at the overall multivariate pattern to predict executive function in unseen data. So what Zaishu did is he used split-half ridge regression with nested parameter tuning to look at unseen data and predict executive function that was measured out of the scanner. And what you can see is we can predict executive function from the complex pattern of functional topography pretty well. R values above 0.4 in both halves of the data. However, then, despite not finding any associations with age in the overall total network representation, we tried the same multivariate pattern regression technique to predict age. And somewhat to our surprise, we could predict it even better. And this is, again, controlling for known confounds like motion artifact, which I'm not talking about today. Sorry, guys. And we can predict age even better. R values in split halves above 0.7. And even though the overall network representation didn't change with age, what's driving this very high predictive performance is a complex pattern of network differentiation that you can see when you look at the feature weights of these models. And what you can see is that some networks get stronger, other networks get weaker, and in particular, for example, the default mode is becoming less represented in the frontoparietal network, whereas the frontoparietal network is becoming more represented specifically at the border. And what we think is happening is these networks are differentiating and becoming sharper as kids get older. This is actually convergent with that process I was talking about earlier of network segregation that we know from group atlas network data. So as a next step, we can look at all the feature weights to see what's driving these predictive performance in these models. And in both the model that predicts age and the model that predicts executive function, the most heavily weighted features were found in a higher order association cortex in the frontoparietal and ventral attention networks that are the most variable in individuals. So the contribution weight was linear related in both models to network variability. So to put this together, what we have is we have highly variable functional anatomy in higher order association cortex, especially the frontoparietal and ventral attention systems, that reconfigures in development and predicts executive function. As a final step, we wanted to understand if there's any fundamental properties of the cortex that constrain this variability. And so what we did is we used a very, very statistically conservative spatial permutation procedure that preserves the spatial covariance structure of this data. This was implemented by Aaron Alexander Block at Yale. And what this allows us to do is to test whether network variability aligns with evolutionary expansion, myelin content, and cerebral blood flow. And what we found is that this network variability, which we know drives reconfiguration and development and is associated with the individual differences in executive function, is associated with higher evolutionary expansion, lower myelin content, and higher CBF. So just to summarize, what we found is that executive function is associated with greater representation of control networks in the cortex, and that age can be predicted from this complex pattern of network differentiation. Furthermore, in both these models, the features that are most predictive are present in higher order association cortex within the frontoparietal cortex, and that these same highly variable parts of the cortex have expanded over evolution, are lightly myelinated, and receive a disproportionate amount of blood flow. These results, when you step back, are actually quite consistent with an evolutionary model put forward by my former mentor Randy Buckner with phenocrenin, which suggests that in evolution, association networks become untethered from this rigid, hardwired somatosensory cortex, and that this may allow for more flexibility, more individual differences, more adaptation and development. But it comes at a cost, too. It comes at a cost of higher metabolic costs and also potentially a higher vulnerability to neuropsychiatric syndromes. So the obvious next step is to understand how these highly individualized networks associate with things that I care about clinically, like neuropsychiatric syndromes that start in development. I'm going to stop by thanking the huge group that makes this possible, in particular the students who do all of the work. Cedric Shia, who will be leaving us soon, Zai Shu, who is here and is doing fantastic work. I want to thank Raquel, who's the overall PI of the PNC, Damon, who's worked with us on the functional parcellation project, Danny Bassett, who will give a keynote right after that, who's the best collaborator a human could ever have, Taki Shinohara in statistics, who's back there, Yong Fan, who developed the NMF technique, all of our funding from NIMH, and all the people who participate in our studies. Thank you.